Swayam Prabha. Digital India. Educated India.
Hello learners, today we are going to discuss about the phylogenetic trees from the module Evidences of Evolution under the paper Evolutionary Biology. Now under this module we have discussed so far about various evidences of evolution, particularly focusing on paleontological and molecular evidences as per our curriculum, and this is the last lecture under this module where we are particularly going to focus upon the phylogenetic trees. I'm Dr. Sudhir Verma, working as Assistant Professor in Department of Zoology, Deen Dayal Upadhyay College, University of Delhi. Now under this lecture, we are going to learn about the interrelationship between the taxonomy and phylogeny branches of biology. Then we'll focus upon the phylogenetic trees. What are their types and the terminology that is being used for the depiction of phylogenetic trees. Later on, we'll see how the trees are being constructed, the various methods that are available for construction of phylogenetic trees, which are broadly classified under two categories, that is distance based and the character based. Then we will focus upon the evaluation of the trees that we have constructed, how reliable, how robust that tree is. We'll discuss about the uses and the limitation of the phylogenetic trees. So these are some of the subheads under which we'll cover today's lecture. Starting with the interrelationship between taxonomy and phylogeny. Now taxonomy or systematics are the branches of biology that deal with classification and naming of organisms according to certain rules. Depending upon the parameters that are used for the classification, there are two broad categories. One is called as phonetics and another one is called as cladistics. Let us see what are their differences. Though both are taxonomical methods of classifying organisms, they do differ in their certain characters. For example, if you see a definition of phonetics versus cladistics, phonetics is a method of classifying organisms based on the structural and the morphological characteristics that are observable. So whatever you see, the characters which you can see morphologically, based on that we try to classify the organisms under the phonetics. Whereas in cladistics, we classify organisms based on the ancestry and their evolutionary relationship. So these are some things which we do not see as such. The characteristics which are considered under phonetics are mainly their structures, their morphology, their locomotory patterns, etc. Whereas in case of cladistics, these are the evolutionary relationships, the genetic similarity and the ancestry. The use of trees to describe relationship under phonetics is mainly the phonograms, whereas under cladistics these are the cladograms or the phylogenetic trees. The accuracy and reproducibility both are low in phonetics as compared to cladistics, which are highly reproducible and highly accurate. Now we are going to discuss or focus particularly on these cladograms and phylogenetic trees. That means we are going to discuss the taxonomy with relation to evolutionary history. That means taxonomy and phylogeny connections through the phylogenetic trees. The evolutionary relationships of a species or a group of species can be used to build taxonomic groups. The evolutionary history of a species or a group of species is called phylogeny, and thus a phylogenetic tree is a hypothesis that depicts the evolutionary relationship among group of organisms, and thus taxonomy should reflect the phylogeny. So if you are going to classify a group of organisms, if you are going to depict their relationships, it has to reflect their phylogenetic relationship, their evolutionary relationship or history. Now let's move to the phylogenetic trees. Phylogenetic trees are nothing but a visual representation of the relationship between different organisms, showing the path through evolutionary time from a common ancestor to different descendants. That means visually when you show the relationships, particularly the evolutionary relationship between different organisms starting from their common ancestor to the present forms or their descendants, that is called as phylogenetic tree. The phylogenetic trees are hypothesized reconstructions of evolutionary history. Remember, these are hypothesized reconstructions. We do not know exactly what has happened in the history, but they are very much reliable to show what probably would have occurred during the evolutionary history. Now, one of the example has been shown here of a phylogenetic tree. Here you can see there is a root node which represents a common ancestor for these three different taxa, which are A, B and C here. Now from this there is a bifurcation which is leading to taxon C, which is acting as a outgroup for taxon A and B. So from this diversion C got separated. From here there is another internal node which is giving rise to taxon A and taxon D, sister taxons. So this is one of the very simple example of a phylogenetic relationship in a tree. Remember, the trees can be represented in parenthesis notation as well. Each set of parentheses represents a branch point or a bifurcation, and the comma represents left and the right lineages. Now taking the same example which we discussed in the previous slide, if this is the tree, it can also be simply shown as this parenthesis notation. That means taxon A and B represents a branch point, a bifurcation, and a comma in between then represents left side and the right side. So if you see here, A and B are two different taxons separated by a comma, which means that they are on the left side or right side, and a parenthesis which is covering these two, which means they are bifurcated, and further by a comma they are joined with a C, that is here. So this is how we show a phylogenetic tree, simple phylogenetic tree, using a parenthesis notation. The trees can be shown in a horizontal arrangement, vertical arrangement or a diagonal arrangement. Now what you see here in this slide, it is the same type of tree, but it has been shown under three different depictions. So representation wise they look different, but if you focus, they are the same tree. So here if you see, MRCA is nothing but the most recent common ancestor. So from this most recent common ancestor, there is a bifurcation leading to further bifurcation and leading to all these recent taxa. Now this is a horizontal depiction. Here if you see, the same thing has been shown in a vertical depiction, and here it is the diagonal representation. So A and B are joined together, A and B joined together, A and B joined together here as well, and then they are joined with C and D, joined with C and D here, joined with C and D here. So if you see, both the all the trees are same, but representation wise they do differ. Also, if we rotate the structures using one of the branch point as a pivot, we do not change the relationship in a phylogenetic tree. Now, different depictions of the same phylogenetic tree has been shown here. All of these depict the same relationship between the organisms, that is A, B, C and D. Now, if you focus here, let's say C and D are being joined by this branch point. If you rotate it, it may become like this. If you rotate the branch which is joining B with C and D, it may become like this, and so on. So all these trees are nothing but the same type of tree which has been rotated using certain branch points, but the evolutionary relationship between the taxon has not changed. Below here, it's nothing but the diagonal relationship of the same trees only. There is also something called polytomy. In the previous trees, most of the time you have seen this kind of bifurcation, but it is not necessary that we'll always have this bifurcation only. There can be a phenomena of polytomy as it is shown here in the P, Q and R. Poly means many and tomy means cuts. So there are many cuts here. Why this happens is because we do not have information sufficient enough to decide their order, to decide their branching order. So that is why they are being shown all together joined with one branch point. The phylogenetic trees show the relatedness. Now what do we mean by relatedness? Let's see here in this tree. In the phylogenetic trees, if two species are more related, they will have more recent common ancestor. Or in other words, if two species are more related, they have a more recent common ancestor, and less related if they have a less recent common ancestor. Now if you see here, starting from this, that is the ancestral species, and these are the present day species. So for A and B, this is the most recent common ancestor. For AB and CD, this is the most recent common ancestor for B and C here. So they show the relatedness and the time history as well. Let us try to understand these phylogenetic tree with an example. In this table, some characters are shown here in the rows, and in the columns there are different organisms: lamprey, antelope, bald eagle, alligator and the sea boss. The X here in different columns represent that the presence of that particular character in that particular organisms. So for example, lungs are present in antelope, eagle, alligator, but not in bass and lamprey and so on. So here you have the characters and here you have the organisms. Now taking this example of characters and organisms, we can create their relationships like this tree. For example, let us start with the most ancestral character. That means the character which is present in all the organisms. Here if you see the notocode which is present in all the organisms, so probably the ancestor had this notocode from where the bifurcation started. So from here there is a bifurcation started based on the jaws. So if you see lamprey, it did not have the jaws except all other organisms which are having the jaws. So from the character which was present in all, we are moving to a character which is absent only in this organism but present in all other organisms. So from here the lamprey can be put out or separated out, and then based on jaws again we can have a classification or the bifurcation based on the lungs. Here we are going further down. After jaws we are coming to lungs which is present in three organisms here, not in the boss and lamprey, and so on. You keep on building the tree like this. But if you have these kind of trees, can you tell about what is the amount of change in characters? That means how much or quantitatively how much that character has changed throughout the evolutionary history. Can you tell about the amount of time? That means the time spent in the history or evolutionary history that it has spent or undergone the change. based on these kind of questions, there are different kind of representations or the type of trees. For example, they can be dendrograms. Dendrogram is a broad term for diagrammatic representation of a phylogenetic tree. Any diagrammatic representation of a tree is called as dendrogram. The term has been derived from the Greek words which mean tree from the word dendron and drawing or mathematical figure from the word gramma. So any kind of diagrammatic representation can be termed as a dendrogram simply. Whereas another term is there which is called as cladogram. It represents a branching pattern, that is, its branch length do not represent time or relative amount of character change, and its internal nodes do not represent ancestors. The word cladogram has originated from the Greek word which means branch from the word clados, and gramma means the character. It can be based on the morphological data, just like the skulls, the type of skull present, notochord whether it is present or absent, and so on, or the molecular data in terms of DNA or amino acids. It can be a vertical cladogram or it can be a horizontal cladogram. So here the point to be remembered is the branch lengths are not representing time or the amount of character change. Moving further, there is a type which is called as chronogram. Chronogram is a phylogenetic tree that explicitly represents time through its branch length. So here it is not the random length of the branch, but the length of each branch represents time. So if this is the recent time, the present time, it is being represented by a 0. So here, that is, this location is at 1, this is 2, 3. So based on that, you can predict the time, relative time particularly, when this divergence took place. So here you can get an idea about the amount of time that it has changed, and this is called as chronogram. Yet another category is called as phylogram. Phylogenetic tree that has branch lengths proportional to the amount of character change is called as phylogram. So here the branches, the length of the branches represents amount of character that it has changed. So if it is a short length branch, that means it has undergone smaller change as compared to a longer branch, which means it has undergone a higher amount of change. So in phylogram, the branch length is representing your amount of character that has changed. Now, three broad categories we can have of the phylogenetic trees based on the branch length. If the branch length does not convey any meaning, that means it is simply a cladogram. If the branch length represents the genetic change, the amount of change of character, it is called as phylogram. Whereas in case of ultrametric trees, the branch length represents the time. So based on whether the branch length is depicting time or genetic change, or it is just showing the relationship without mentioning the time or the amount of character change, they can be classified as ultrametric trees, phylograms, or cladograms respectively. Now another point I would like to tell you here is the parenthesis notation which we discussed initially. They can have both the labels and the distances as well. So here you can see for example, C and B here. C is 1, B is 6. This is the relationship of the distance as well. So not just labels C and B, but the distances also are being shown here. That is also possible in the parenthesis notation. Now let us see elaborately how a tree is being labeled. What are the different terms that we use when we represent a phylogenetic tree? If a tree is rooted, that means if it has a root to it, just like a normal tree or the common ancestry, this is called as root. This is called as hypothetical taxonomical unit or HTU, which is hypothetical taxonomic unit. We do not have it now, but we hypothesize that this is the common ancestor from which the different branches have emerged. There is something called internal node and there is something called as external nodes. Internal nodes is like here, which is a ancestor for all these, whereas external nodes are the taxons which are present here. Now these are the taxons which are called as operational taxonomical unit in contrast to hypothetical taxonomic unit. These are the OTUs. There is an internal branch and there can be an external branch. Just like internal node, we have external nodes also, and these are the taxons which are having a common ancestor. So this represents a particular clade. Now within a tree there can be three types of relationships. One is called as monophyletic, another one is called as paraphyletic, and yet another one is called as polyphyletic. Let us discuss what are these. In this same tree which is being shown here, this is one type of grouping shown here. Now in this tree, in this grouping 1, there are 7 species B to H, and it represents a monophyletic group, which means a group which is made up of an ancestral species, which is B here, and all of its descendant species. So a common ancestor and all its descendant species, if they are depicted here all together, it is called as monophyletic group. Only monophyletic group qualify as legitimate taxa derived from cladistics. Now compare this monophyletic with this paraphyletic here, which is shown in this grouping 2. Grouping 2 does not meet the cladistic criteria. Why? Because it is a paraphyletic grouping, which means that it consists of an ancestor, which is A in this case, and some but not all of its descendants. Whereas in monophyletic, the common ancestor and all its descendants were grouped together. In case of paraphyletic, there is a common ancestor, but all its descendants are not shown. Only some of its descendants are grouped together. So in this case, though B and all its descendants are also coming from A only, but only we are discussing the descendants coming onto this group. So this is called as paraphyletic groups. The polyphyletic groups, as shown in group 3 here, also fails a cladistic test. It is polyphyletic, which means that it lacks the common ancestor. For example, here these are the derived groups or descendants, but it does not include the common ancestors here. Furthermore, a valid taxon that includes the extant species G, H, J and K would necessarily also contain D and E also here, which are also descendants from A. But in this case, it is not being included here. So again, this is not a true cladistics. So based on which groups you are covering in a group, whether you are including the common ancestor or not, whether you are including all the descendants or some of the descendants only, based on these characters, we decide whether it's a monophyletic group or paraphyletic group or the polyphyletic groups. The trees can also be differentiated as a rooted or unrooted tree. A rooted tree has the ancestry relationship. It shows the ancestry relationship, whereas unrooted phylogenetic tree, it only shows the relatedness of the organisms. It does not talk about the ancestral relationships. Rooted trees do contain a root, internal nodes and the leaf nodes, whereas unrooted tree contains leaf nodes and internal nodes only. There is no root present in this. The rooted tree contains related organisms as well as an extra distantly related organisms for the root, whereas the unrooted trees contain only the related organisms. The phylogenetic trees which are rooted, they do show the recent common ancestor, which is the root for each descendant. It is not present in the unrooted tree. Each root to a node shows the evolutionary path here, whereas it does not show the it does not specify the evolutionary path if it is an unrooted phylogenetic trees. A rooted phylogenetic tree shows the ancestral state of the organisms or genes at the bottom to the terminal branches, whereas unrooted tree does not show the ancestral states. So if you see broadly, they do differ in terms of whether a root is present or not. A root basically means the common ancestral form of the descendants. So here you can see this is an unrooted tree which shows the interrelationship of A, C, B and D here, but it does not talk about the ancestry. Here in the second and third part, there is a midpoint rooted tree and an outgroup rooted tree. In a midpoint rooted tree, you can see A and C and the another clade which is B and T. They do have a root, they do have a common ancestor. Whereas in outgroup rooted tree, you can see a D species is there, which is totally different, totally unrelated from this A, C and B. So this is, but they do also have a root here. So this is how a rooted tree differs from an unrooted tree. Now let us move to the construction part of phylogenetic tree, when we have got a brief idea about what are the types of trees, how do we interpret the trees, what are the terminologies that we use. Based on that, now let's move forward how we construct the trees. Now there are 4 basic steps for construction of any type of phylogenetic tree. First, we need to identify and acquire the sequences of homologous proteins or DNA. Once we have this, we need to align these sequences, this molecular alignment, which is a multiple sequence alignment, because we are going to discuss the evolutionary relationship of many organisms or species. So it will lead to multiple sequence alignment as compared to pairwise alignment, which is also there when we compare 2 sequences only. Then once we compare these, we do estimate the tree from the aligned sequence. So we hypothesize their relationship in the form of a tree. And then finally we present the tree and interpret the interrelationships of various taxons which are present. So starting with the first step, which is the identification and acquisition of the homologous proteins or DNA sequence. So we need to select one of the protein or gene or DNA sequence which is homologous. You must be aware of the homologous sequences which we have discussed in the previous lectures. What you can use for acquiring these sequences is you can go to various databases and you can retrieve the sequences from there. Some of the databases you might be aware of is NCBI, DBJ, EMBL and so on. So you can retrieve your nucleotide sequence or protein sequences from there. Once you have all these sequences, you need to align them. You need to see their homology, and you can do multiple sequence alignments or MSA. And for that also you do have various softwares available. 2 of the very prominent and very widely used softwares are the Clustal and the Muscle. Now I'll show you simply their web page. So this is the Muscle software which appears like this, and this is the Clustal Omega, which you can search on internet, and you can use these softwares for your multiple sequence alignment. Now in phylogenetic tree, when you align the sequence for multiple sequence alignment, this is something which you see. What you can see here on this side, this is a nucleotide sequence alignment, whereas below this is a protein sequence alignment. Now what you can observe here is there are certain colors used here. and there are certain symbols which are being used here. Now, what do these colors and symbol mean? Each residue in the alignment is assigned a color if the amino acid profile of the alignment at that position meets some minimal criteria specific for the residue type. So depending on the category of amino acids, what are the residue at that position and so on, there are some default color codings which Clustal use, and you can, based on the color, you can predict what kind of amino acids are there. Similarly, there are certain symbols which are used, which are called as consensus symbols. They do denote the degree of residue conservation. For example, if you compare 3 or 4 species sequences and at base you see an asterisk has been marked after multiple sequence alignment by the software, it indicates the positions which have a single fully conserved residue. Whereas a colon indicates conservation between groups of strongly similar properties, whereas a dot or a period indicates conservation between groups of weakly similar properties. So depending on the properties and their conservation, different symbols are being given at the base of multiple sequence alignment of the bases sequences. Now let us quickly move to estimation of trees from the aligned sequences. There are two major methods. One is called as phonetic methods, which are based on the distances, and thus they are also called as distance based methods. The prominent examples are UPGMA, that is unweighted pair group based on arithmetic mean. Another one is NJ, that is neighbor joining method. There is another branch which is called as cladistics method based on the characters, or they are also called as character based methods. The examples include MP, that is maximum parsimony, or ML, which means the maximum likelihood.